IB Bio, DNA Structure and Replication Part 2, will focus on the detail of DNA structure. DNA Structure and Replication Part 3 will focus on the details of DNA replication. The essential ideas are genetic information in DNA can be accurately copied and can be translated to make the proteins needed by the cell. As well, the structure of DNA is ideally suited to its function. The replication process was immediately clear to Watson and Crick upon the elucidation of the structure of DNA in 1953. The breaking of the hydrogen bonds so that DNA could unwind exposes the template strands so that they can be copied using complementary base pairing rules. As we will understand later, the construction of the new strands occurs in opposite directions. Watson and Crick could not have predicted this in 1953. That the two new chromosomes are composed of one template strand and one newly built strand means that replication is semi-conservative. The first IB syllabus statement to kick off this movie, a statement you've already seen in movie part 1, is state that DNA replication is semi-conservative and depends on complementary base pairing. Again, in this image, you can see each parental strand serving as the template for the building of a new strand. This is semi-conservative replication. Base pairing rules ensure that once replication is finished, the two new molecules are identical. Remember that replication happens during the S phase of interphase, and the two molecules would be held together by the centromere until pulled apart during cell division. The synthesis of DNA from a DNA template is called replication. Keep the terms replication, transcription, translation distinct. Transcription is the synthesis of RNA from the DNA template, whereas translation is the synthesis of a polypeptide from an RNA template. Here is the Watson and Crick central dogma once again, but notice that I've added RNA to DNA. Now this is a bit unusual, but RNA viruses, such as HIV, have an enzyme known as reverse transcriptase that copies RNA to DNA. We'll come back to look at this enzyme when we study genetic engineering. The double helix is composed of two strands of DNA with complementary base pairing toward the center of the molecule. Always a purine paired with a pyrimidine, a purine paired with a pyrimidine. You can see the opposite orientation of each strand with respect to the other. This is referred to as anti-parallel. Now, I've already mentioned that each strand is replicated in opposite directions. If this strand were to elongate, new nucleotides would be added here. If this strand were to elongate, new nucleotides would be added here. The covalent links between nucleotides would be here and here and here, between the number 3 carbon of the sugar and the phosphate group. These are the 5 to 3 linkages. This is a simple image that does a nice job of displaying the semi-conservative nature of replication. The red parental strands here and here serve as a template for the building, the synthesis of the blue daughter strands. Remember that replication occurs during interphase prior to cell division. A DNA molecule unwinds and each parental strand serves as the template for the building of a new strand so that the two molecules of DNA that are formed are each identical to the other due to base pairing rules. Remember that the two new molecules of DNA are called sister chromatids and would be held together by a centromere, the X-shaped chromosome, as we know it. The two new molecules of DNA following replication are seen here and here in an electron micrograph, and these are known as sister chromatids, and they're held together by the centromere prior to cell division when the sister chromatids are pulled apart in mitosis or meiosis II. Now there are two IB syllabus statements that can be mentioned here. Eukaryotic chromosomes are linear DNA molecules associated with histone proteins. Nucleosomes help to supercoil the DNA. Chromosomes are composed of DNA and protein histone protein. Recall that in prophase, the chromatin condenses in two chromosomes. Condensation is often called supercoiling. DNA is wrapped 2.5 times around 8 histone proteins, and this is called a nucleosome. You can see nucleosomes in greater detail here. It's the DNA wrapped 2.5 times around 8 histone proteins. Nucleosomes help to supercoil chromosomes, and they help to regulate transcription. Transcription is the synthesis of RNA from a DNA template, and when the DNA is wrapped around histone proteins, it's not available to the enzymes of transcription. Here are the IB syllabus statements about nucleosomes. Nucleosomes help to supercoil the DNA. Nucleosomes help regulate transcription in eukaryotic cells. Here is a diagram of the prophase chromosome condensed, supercoiled, and replicated. This is a nice image to study to understand the 1-to-1 ratio of purines, A and G, to pyrimidines, T and C, with the hydrogen bonds that hold the bases together, that hold the two strands of DNA together. Now, can you identify a single nucleotide? Here is one, just like that. The phosphate group, the 5-carbon, the 3-carbon, and the covalent bond that links nucleotides here and here and here. On the other strand, here's your phosphate group, the 5th carbon, here's the 3-carbon, and this is the covalent bond that links this nucleotide to this one. This is a covalent bond, this is a covalent bond, and this will be one when a new nucleotide is added here. Here's a close look at nucleotides, specifically the purines, pure as gold, A and G, and the pyrimidines, C and T. Once again, let's do some carbon counting. This is the number 1 carbon, and it's bound to the base. This is the number 2, the 3, the 4 carbon is here, and the 5th carbon is here with the phosphate group attached. New nucleotides are added to the number 3 carbon only. Let me say that again. New nucleotides are added to the number 3 carbon only. Here are the purine, pyrimidine base pairs, A with T, G with C. Hydrogen bonds hold the base pairs together, holding the two strands of the double helix together. In this image, we can see a single nucleotide here and one here. Notice the anti-parallel orientation of the strands. The covalent links between the 3 carbon of one nucleotide and the next nucleotide is here and here and here, and on the other side here, here, and here. This is the 3 end of this strand because this is the 3 carbon, and this is the 5 prime end of this strand because the 5th carbon is here. On this strand, this is the 3 end, this is the 3 carbon, and the 5 carbon is here. Notice the slight mistake by the author of this image. The notation of the 5 prime is given here, and that's not located quite right. It should be here. So here's the IB syllabus statement that moves headlong into the details of replication. Explain the process of DNA replication in prokaryotic cells, including the role of deoxynucleoside triphosphates and polymerases that can only add nucleotides to the 3' end of an existing nucleotide. The nucleotide monomer required in the building of a new DNA polymer comes from a nucleoside triphosphate. The term nucleoside is used instead of nucleotide because this molecule has 3 phosphates, not 1, as would be found in the nucleotide. In the polymerization process of DNA replication, the nucleoside triphosphate is broken. 2 phosphates are released to form the nucleotide. The nucleotide is added to the growing DNA polymer. The energy required for this anabolic polymerization, a dehydration synthesis, comes from the nucleoside triphosphate. Just like ATP, these triphosphate bonds have free energy to give to the replication process. Let me say it again. The energy contained in the nucleoside triphosphate provides the energy necessary for the anabolic polymerization of DNA, the replication of DNA. In this image of a deoxyribonucleoside triphosphate, I've indicated the two phosphates that are cleaved off to form the nucleotide. The energy in the phosphate-phosphate bonds provides the energy necessary to take the nucleotide and add it to a growing DNA polymer. But there's been something lurking beneath the surface that's very important for you to attend to, and that is state that DNA replication occurs in a 5' to 3' direction. Review the next few slides very carefully. In this image, here is the template, the parental strand, and the growing strand is here. Here's a nucleoside triphosphate arriving to be added to the growing DNA strand. It adds as a nucleotide by releasing two phosphates, seen here, moving left to right. The new chain grows in a 5 to 3 direction. The nucleotide is added only to the 3 carbon of the existing chain. This is the 5 end, and this is the 3 end, and the new DNA strand is built in this direction, a 5 to 3 direction. If this strand of DNA were to lengthen, replicate against a template, it would grow in a 5 to 3 direction, this direction. New nucleotides would be added to the 3 carbon only. If this strand were to lengthen, replicate against a template, it would grow in a 5 to 3 direction, this direction. New nucleotides would be added to the 3 carbon only. The covalent links between nucleotides can be seen well here, 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 and here. I've come back to this image to strengthen the concept that as the DNA unwinds and new strands are constructed against template or parental strands, they're constructed in opposite directions. Let's say, by way of example, 5 to 3 here and 5 to 3 here in the opposite direction. This is the image proposed by Watson and Crick in 1953 as their theory for replication. Their theory was correct except for the direction of replication, which by these arrows indicated that both of the new strands are built in the same direction. You now know that one strand is built in this direction while the other is built in this direction. Once again, new nucleotides are added onto the 3 carbon here, so this strand would grow in this direction, 5 to 3. New nucleotides would be added onto the 3 carbon here, so this strand would grow in this direction, 5 to 3. The IB syllabus statement of relevance is DNA polymerase links nucleotides together to form a new strand in a 5' to 3' direction using the pre-existing strand as a template. Here is another image displaying the 5 to 3 direction of DNA replication along a template strand. New nucleotides are added onto the 3 carbon end here, so this strand would grow in this direction, 5 to 3. To foreshadow the next movie in the series, we can see DNA replication occurring in a 5 to 3 direction here against a template strand. The enzyme that polymerizes DNA during DNA replication is DNA polymerase. In prokaryotic cells, the enzyme is better known as DNA polymerase III. DNA polymerase uses the template strand and base pairing rules to construct the new DNA strand 5' to 3' direction. In comparing DNA replication to transcription and translation, in replication, DNA polymerase III catalyzes the synthesis of DNA from a DNA template. In transcription, RNA polymerase catalyzes the synthesis of RNA from a DNA template. And in translation, the ribosomes, large enzymes, catalyze the synthesis of a polypeptide from from an RNA template. And that brings us to the end of DNA structure, DNA replication part 2. In DNA structure, DNA replication part 3, we will look at the details of DNA polymerase, as well as 4 other enzymes important to the replication process.